Have you ever tried to find a really tiny, subtle detail in just a massive, overwhelming sea of information?
Oh, yeah. It's incredibly frustrating.
Right. Like, picture standing in the middle of a screaming, sold-out rock concert. The bass is just rattling your ribs, and you're trying to hear, a single specific whisper from across the stadium.
That is a perfect analogy because your brain knows the signal is out there somewhere, but, the sheer volume of the surrounding noise just completely saturates your perception. You just can't filter it out.
Exactly. And that scenario isn't just a minor annoyance. It actually represents one of the most critical bottlenecks in modern medical research right now.
Yeah, absolutely.
So today we are bringing you, the listener, along on a mission. We are diving into a technical report published in Nature Cell Biology about this groundbreaking new computational framework. It's acting like a super sleuth in the world of spatial biology.
And it's an incredibly powerful tool. It's called CoCo-ST.
Right, which stands for Compare and Contrast Spatial Transcriptomics. The developers designed it specifically to find those hidden, microscopic whispers of early disease that, honestly, other state-of-the-art computational tools just completely miss.
They just gloss right over them.
Okay, let's unpack this. To really understand how CoCo-ST changes the game, we need to know what game we are actually playing. As an audience tuning into this deep dive, you likely already know that traditional transcriptomics gives us the what, right? By analyzing the RNA, we know which genes are active. But spatial transcriptomics, that gives us the where.
Exactly. And that spatial mapping is the revolution we've been living in for the past few years. We aren't just blending tissue into a smoothie anymore to see what's inside.
Right, the old bulk RNA smoothie approach.
Yeah. Now, we are looking at a bustling city map, and we can see exactly which houses have their lights on. But here's the major problem with the current state-of-the-art dimension reduction technology. Methods you've likely worked with, like principal component analysis, or SPAGE-ECN, or Bayes space.
The heavy hitters.
Right, the heavy hitters. When these algorithms look at that city map, they are mathematically programmed to hunt for the biggest, most obvious variations in the data. In data science, we call these high-variance structures.
They're looking for the massive shifts in the eigenvectors, right?
Precisely. They want the big structural differences.
So if we think about our city map analogy, a high-variance structure would be like a giant flashing neon sign. In a biological sense, that's an advanced, obvious tumor margin where the gene expression shifts, dramatically compared to the surrounding normal tissue.
It's impossible to miss.
But what about the low-variance structures? Those would be like a tiny flickering light bulb in a single window down a dark alley, like an early precancerous cell.
Right.
And because traditional algorithms are so heavily weighted to capture those giant neon signs, they just treat that single flickering bulb as background noise. They essentially smooth it right out of existence.
Which is a massive, massive blind spot when you consider the actual biology of carcinogenesis, how cancer develops.
Because it doesn't just happen overnight.
Exactly. Tumors don't just spontaneously appear as giant, flashing neon signs. They evolve from very subtle, localized miscommunications between just a handful of cells. They start as these tiny, isolated, early adenoma regions.
Just a few rogue cells whispering to each other.
Yeah. And if our best analytical tools can't see the flickering light bulb because the global variance of the rest of the healthy tissue is too loud, well, we're entirely missing the crucial window to understand the disease at its inception. We're only catching cancer when it's already screaming at us.
Wow. Okay, so I want to do a quick recap here for you listening, just to lock this in. First, spatial transcriptomics maps genes to exact locations. Second, current tools really only see the obvious loud patterns, the high variance stuff. And third, the subtle early diseases get completely ignored as noise.
That's the baseline problem in a nutshell.
So here's a question for you, the listener. Think about your own field of work. How often do the loudest problems get all the attention, while the really subtle, critical warning signs just get completely ignored until it's too late?
It's a universal problem, really.
It is. So if traditional tools are blinded by the neon signs of advanced disease, that brings up the obvious engineering challenge. If the biological whisper of an early tumor is just too quiet to hear over the rock concert, and we can't magically amplify that whisper without distorting all the other data, we have to approach it from the other side.
Right. We have to quiet the room.
We have to shut down the concert entirely.
And that operational pivot, that is the core genius of CoCo-ST. It operates on a mechanism called graph contrastive feature representation.
Graph contrastive feature representation. Break that down for us.
So rather than taking a single piece of suspect tissue and just trying to run clustering algorithms to find anomalies, CoCo-ST actually requires two distinct inputs. It takes a target sample, which is the tissue we suspect might contain some hidden abnormality.
Okay, the target.
And it pairs it directly with a background sample, which is a known, healthy baseline tissue.
So it's looking at both simultaneously. Let's open up the hood on the objective function here, because the math in the paper is where this gets really elegant. How exactly is the algorithm comparing these two inputs without just washing out all the data?
Well, it starts by building a mathematical graph of the local geometric structures for both the target and the background. It uses the spatial coordinates to define localized neighborhoods of cells, essentially creating an adjacency matrix.
Okay, so mapping who is next to who.
Right. Then it seeks to maximize the spatially coherent features that are unique to the target data set. But, and this is the crucial part, while it is maximizing the target's variance, the objective function actively penalizes or mathematically subtracts the variance of the features that are shared with the background data set.
It literally subtracts them.
Yes. There is a specific variable in the equation, the contrastive parameter, which they call eta. And eta basically acts as a tuning dial for this trade-off between the target and the background.
Okay, so by firmly defining what is normal from the background, the algorithm is forced to focus only on the abnormal in the target. I love this. It's exactly like how noise-canceling headphones work.
Oh, that's a brilliant way to put it.
Right. The headphones have a little microphone that measures the constant ambient noise of the airplane cabin. That's your background. Then, the computer chip inside generates an exact inverse sound wave and subtracts it from your audio feed. And suddenly the drone of the jet engines just vanishes, and you can perfectly hear the quiet podcast you're trying to listen to. CoCo-ST is doing the exact same thing, but applying that inverse wave to biological geometry.
What's fascinating here is that your analogy maps perfectly to the matrix algebra in the paper. The objective function literally centers on a subtraction sign. And it's the local variance of the target graph minus eta times the variance of the background graph.
It's just beautifully simple when you think about it.
It really is. By subtracting the high-variance normal tissue patterns that are abundant and shared across both samples, the loud global noise is canceled out entirely. The massive eigenvectors of the healthy tissue are mathematically neutralized.
And what is left behind?
The unique low-variance local structures, the whisper that was there all along.
Man, that is elegant. Let's pause for another mini recap. Point 1, CoCo-ST uses two inputs, a target and a background. Point 2, it compares their local variance graphs. And point 3, it mathematically subtracts the normal background to highlight the unique disease structures.
Spot on.
And a quick question to throw to you, the listener. Consider how you learn a new concept. Do you sometimes find that you understand what something is much better when you first define what it isn't?
Contrast is a powerful learning tool.
It really is. Now, the math is incredibly elegant, but, theory is one thing. How does this actually hold up when you introduce the messy, chaotic reality of biological tissues? Because a lot of algorithms look great in a Python environment, but they just fall apart in the wet lab.
Oh, definitely. The researchers obviously knew that, so they had to benchmark this against the heavyweights of the industry.
And did they?
They benchmarked it aggressively. They threw CoCo-ST into the ring against 10 state-of-the-art algorithms. We are talking about industry standards like Seurat, Mephisto, Stegate, Giotto, all the big names.
Okay, 10 algorithms. What was the setup?
For the test, they used a mouse carcinogenesis model. They took a normal mouse lung labeled MLP1 to serve as the background baseline. Then they took an abnormal mouse lung, MLP6, and fed that in as the target.
And what was the defining difference in the output? What were they looking for?
Well, in the target tissue, there was this tiny, highly localized hotspot region. A human pathologist, analyzing the physical histology slide, had manually annotated it as hyperplasia.
Okay, and for anyone not familiar with pathology, hyperplasia is basically a thickening of tissue caused by rapid cell division. It's a classic precancerous state, right? Like an early adenoma.
Exactly. It's that flickering light bulb we talked about. CoCo-ST found it effortlessly. It mathematically isolated and clustered those specific spots on the spatial map perfectly.
And what about the other 10 algorithms?
They completely failed. Every single one of them. They either smoothed the hyperplasia into the surrounding healthy lung tissue, treating it as noise, or they mislabeled it entirely as some sort of technical artifact.
Wait, really? All 10 missed it?
All 10. But let's talk about how we quantify that success, because, we can't just eyeball a map and declare a winner in this field.
Right, we need hard metrics.
Exactly. When we measure how closely an algorithm's clustering map matches the human pathologist's ground truth, we use a metric called the Adjusted Rand Index, or ARI. An ARI of 1.0 would be a perfect, flawless match. CoCo-ST hit an incredible ARI of 0.88.
Wow, 0.88 is huge.
Right. It is. They also used normalized mutual information, or NMI, which measures the amount of information shared between the algorithm's clusters and the true labels. It hit 0.73 there. In the realm of computational biology, these are exceptionally strong markers of accuracy. CoCo-ST significantly outperformed every single alternative tool on the market.
Okay, I have to play devil's advocate for just a second here.
Go for it.
If 10 of the smartest algorithms in the world looked at this exact same data and didn't see a tumor, how do we know CoCo-ST didn't just hallucinate this tiny hotspot? I mean, if you push a contrastive algorithm hard enough with that eta parameter, might it just invent a pattern in the static just to satisfy its own objective function?
That is the exact critical lens the researchers applied to their own work. They fully anticipated that pushback from the scientific community, so they didn't just point to the spatial map and say, hey, trust the math.
Right, you need biological proof.
They went back into the underlying biology of the specific region CoCo-ST identified. They looked at the differential gene expression in that exact hotspot and found 3,498 marker genes uniquely expressed right there compared to the rest of the tissue.
Over 3,000 genes?
Yeah. Specifically, they highlighted a domain-specific metagene profile that heavily featured genes like CTISH, CXCL15, and SLC34A2.
Okay, and those aren't just random letters in the genome. Those have specific, documented biological functions related to tissue remodeling, don't they?
Precisely. Take CloTush, for instance. It encodes for cathepsin H, which is a lysosomal cysteine protease.
Let's put that in plain English for everyone.
In plain terms, it's an enzyme that degrades extracellular matrix proteins. It literally chews up the scaffolding around cells.
Oh, wow. So that is exactly the kind of biological tool a newly forming tumor needs to carve out space and expand into surrounding healthy tissue.
Exactly. It is highly associated with colorectal and lung adenomas. The algorithm wasn't hallucinating at all. It found the physical mechanism causing the hyperplasia.
That's incredible.
And algorithms like Surat or NMF, which mislabeled the region, they completely failed to isolate these crucial marker genes. Because CoCo-ST quieted the background noise, it was literally the only tool able to hear the actual biological truth.
Okay, time for another quick summary. 1, CoCo-ST was tested on mouse lungs. 2, it beat 10 leading algorithms by finding a hidden early tumor that they all missed.
Yeah.
And 3, it proved its finding wasn't a hallucination by identifying the exact marker genes causing the tumor.
A perfect breakdown.
And listener, think about this. Have you ever found a hidden pattern in your own data or your own life that everyone else overlooked just because you looked at it from a slightly different baseline? It changes everything.
It really does. It's a paradigm shift.
Now, finding a hidden pattern in your data when others miss it is basically the holy grail of data science. But here's where it gets really interesting.
Oh, yeah.
If you've ever dealt with large-scale sequencing, you know that finding one hidden tumor in a single, isolated mouse is an impressive party trick. But modern biology doesn't happen in a vacuum. If we're going to cure complex diseases, we have to compare thousands of samples from hundreds of different labs sequenced on different machines on entirely different days.
Which introduces the absolute bane of a computational biologist's existence: batch effects.
Batch effects. Explain that for us.
Batch effects represent technical, non-biological noise. Let's say Lab A processes a tissue sample on a Tuesday using one brand of reagents, and Lab B processes a biologically identical sample on a Friday using a slightly different chemical wash.
Okay, so the biology is the same, but the process varied slightly.
Right. The capture efficiencies change, ambient RNA contamination levels change. When you feed that combined data into an algorithm, the computer will often cluster the Tuesday cells together and the Friday cells together, thinking they are fundamentally different tissue types.
Just purely because of the technical artifacts from the lab.
Exactly. It completely obscures the real biological signals.
It's kind of like trying to compare two identical photographs, but someone put a blue filter over one and a yellow filter over the other. The computer just sees the pixel values and assumes they're completely different pictures, even if they are of the exact same subject.
That is a great way to conceptualize it. Overcoming those color filters is notoriously difficult. But CoCo-ST approaches this dynamically, specifically because of its contrastive learning framework. By constantly referencing back to a shared, normal background baseline, it anchors the data sets. It acts as this ultimate harmonizer across batches.
So they tested this, I assume.
Oh, yes. The researchers tested this by integrating multiple different tissue samples that were heavily distorted by technical variations.
And how do they measure the success of that harmonization? What's the metric there?
They use a metric called KBET, which stands for K-nearest neighbor batch effect test. Essentially, it evaluates how well mixed the different batches are in the final data projection. If the batches remain separated, meaning the technical noise is still dominating, the score is close to 0.
Okay.
But if the batches are perfectly mixed, indicating the algorithm successfully scrubbed the technical artifacts and clustered solely on true biology, the score is 1. CoCo-ST brilliantly blended the samples together, scoring a KBET value incredibly close to 1.
That is a massive operational victory. But wait, the real balancing act here isn't just mixing the data, right? It's mixing the data without accidentally washing away the subtle signals we've been talking about. I mean, if it scrubs the image too hard to remove those color filters, you run the risk of erasing the tiny tumor whisper along with it.
And that is the most remarkable part of their findings. Even while forcefully smoothing out all that global technical noise across multiple lab samples, CoCo-ST still kept the distinct early adenoma regions intact. Because that contrastive parameter, eta, is focused purely on local geometric variances rather than global distributions, so the subtle biological truths actually survive the batch correction process.
That is wild. So it solves the noise problem, it solves the batch effect problem. But there is one more hurdle for any modern bioinformatics tool, and that is scale.
Oh, scale is huge right now.
The field of spatial transcriptomics is moving at just breakneck speed. The mouse lung data we just discussed was from the Visium platform, which looks at spots of tissue, right? Each spot might contain anywhere from 10 to 50 cells. It's a fantastic technology, but we are moving way beyond that resolution now.
We are. To ensure CoCo-ST wouldn't be obsolete in 6 months, they pushed its scalability to the absolute limit. They moved past the spot-level data and tested it on the Xenium Prime 5K platform.
And that operates at true single-cell resolution.
Exactly. At this level, CoCo-ST seamlessly mapped distinct cell types. It was flawlessly distinguishing tiny populations of tumor epithelial cells from the normal bronchial epithelial cells right next to them.
Which completely changes the paradigm. At the spot level, you are essentially looking at a neighborhood and guessing the general demographic. At single-cell resolution, you are knocking on individual doors to see exactly who is misbehaving inside.
Right. But the paper didn't stop at single cells either.
Wait, they went further.
They took it to the absolute current frontier, subcellular resolution. They applied CoCo-ST to Visium HD datasets. We are talking about bins scaled down to 2 micrometers.
2 micrometers. That is significantly smaller than a single cell.
Yes. We are looking at the spatial arrangement of transcripts inside the cell architecture. And at this resolution, the dataset exploded to 9.4 million spatial spots across the tissue.
Okay, 9.4 million individual data points to cross-reference and map. Usually when you hit those numbers, standard dimension reduction techniques like PCA or NMF hit a massive computational wall.
A brick wall.
Yeah. Trying to calculate a covariance matrix for 9.4 million points requires an absurd amount of memory. You're normally looking at operations that need supercomputers and weeks of processing time.
And that is perhaps the most practical, real-world victory of this entire technical report. The researchers ran that massive 9.4 million spot Visium HD dataset on a standard desktop computer.
Kidding, a desktop.
A standard desktop. And it didn't take weeks. It took just over an hour, 4,258 seconds to be exact.
How is that mathematically possible?
It comes down to how that objective function is built. Traditional methods try to calculate the variance of the entire universe of data all at once, which scales exponentially in complexity. But CoCo-ST focuses entirely on local geometric graphs.
Oh, so it's only looking at its immediate neighbors.
Exactly. It calculates the adjacency matrix only for the immediate spatial neighbors of any given point. It essentially functions as a highly optimized sparse matrix operation. It simply doesn't need to hold the entire global map in its active memory to find the local contrast.
Man, you can imagine how that level of computational efficiency democratizes this kind of research. You don't need a multi-million-dollar computing cluster at a major university to do cutting-edge spatial transcriptomics anymore. A lab with a standard desktop and this algorithm can now unify data from entirely different sources, scrub out the batch effects, scale down to the subcellular level, and extract these incredibly subtle biological insights in the time it takes to go to lunch.
It represents a fundamental shift in bioinformatics. We are moving from the era of merely data collection into an era of true knowledge extraction.
All right, final mini recap before we bring this home. 1. CoCo-ST eliminates technical noise between different lab samples, those pesky batch effects. 2. It scales from whole tissue spots down to single cells and even subcellular levels. And 3. It is computationally incredibly fast.
Excellent summary.
So, listener, imagine having a tool that could instantly translate and unify data from entirely different sources without losing the micro details. How would that change your workflow?
It's something every data scientist dreams of.
So if we pull all of this together, the graph contrastive math, the mouse lungs, the batch effect harmonization, the subcellular scalability, what does this all mean? Why is CoCo-ST such a vital stepping stone for the future of medicine?
Because cancer, alongside a host of other complex autoimmune and neurodegenerative conditions, are fundamentally diseases of tissue heterogeneity and spatial dynamics.
They start small.
Right. They are, at their core, diseases of microscopic miscommunication between neighboring cells. And for decades, our inability to isolate and hear those subtle miscommunications, the low-variance structures, meant our medical interventions could really only begin when the disease had already built a massive high-variance stronghold.
When it was already a neon sign.
Exactly. CoCo-ST is a monumental leap forward because it provides researchers and clinicians with a reliable, mathematically proven way to find the disease niches long before they become unmanageable systemic problems. By defining the healthy background, we can finally map the earliest architectural blueprints of disease. It paves the way for the next generation of ultra-early diagnostics.
We finally have the computational tools to hear the whisper over the roar of the concert. Which leaves me with a final lingering thought for you, the listener, to explore on your own.
Let's hear it.
Throughout this deep dive, we've seen how CoCo-ST utilizes contrastive logic. Comparing a specific target against a known background to find the microscopic signals of cancer in a single slice of biological tissue. But could this exact same mathematical philosophy be applied to our personal health data?
Oh, that's an interesting direction.
Think about it. Imagine an AI framework operating on this same contrastive principle, constantly comparing your daily smartwatch vitals, your heart rate variability, your resting temperature, your sleep cycles. Currently, most health apps compare your data against a global average of humanity, essentially looking for the giant, high-variance neon signs of illness.
Right, they just tell you if you're outside the standard deviation of millions of other people.
Exactly. But what if the algorithm used your own historical data as the background baseline? What if it could mathematically subtract the noise of your daily life, your workouts, your stress, your caffeine intake, and spot the subtle, low-variance whisper of a viral infection or a metabolic shift weeks before you even feel the very first symptom?
That would completely revolutionize personalized medicine.
The mathematical foundation for that kind of hyper-personalized anomaly detection is already here. As we've seen today with CoCo-ST, when you know exactly what the background sounds like, there is absolutely nowhere for the anomalies to hide.